oocyte is the female gametocyte. It is surrounded by the thick zona pellucida. This acts to protect the egg and act as a selective filter before fertilization. It only allows sperm to penetrate. Sperm enters and travels through the uterus and up the fallopian tube where it finds the egg and penetrates the zona pellucida. After a single sperm enters, the zona pellucida undergoes a reaction that makes it impermeable to other sperms. The sperm also starts a reaction that will degrade the zona pellucida over time. However, it needs to remain in place for now to prevent early implantation in the uterine tube. The maternal and paternal chromosomes combine and fuse into a single diploid containing 46 chromosomes. This fertilized egg is now called a zygote. The zygote then moves along the uterine tube towards the uterus. While traveling, it undergoes a series of mitotic cell divisions, a process called cleavage, forming several smaller cells called blastomeres. About 3 days after fertilization, the zygote has formed into a group of about 12 or more blastomeres and is now called a morula and enters the uterus. When the morula enters the uterus, it undergoes multiple changes. Fluid moves into the morula, essentially pushing the mass of cells to one edge and forming a fluid-filled cavity, called the blastocystic cavity or blastocele. The fluid causes the blastomeres to separate into two parts. The first part is the outer cell layer, called the trophoblast, which becomes the fetal part of the placenta. The second part is the inner cell mass, called the embryoblast, which becomes the embryo. The entire structure is now called a blastocyst and begins to expand. The enlargement of the blastocyst, enzymes from the sperm, and the secretions in the uterus begin to degrade the zona pellucida. The blastocyst has now completely shed the zona pellucida. The trophoblasts now expose little microvilli. The endometrium epithelium contains mushroom or balloon-like protrusions, called penopods. The microvilli on the trophoblasts adjacent to the embryoblast loosely attach to the penopods on the endometrial epithelium. The blastocyst is now essentially stuck to the endometrium. The zygote now needs to form a more secure connection to the endometrial epithelium. For this to occur, the trophoblast or outer cell mass differentiates into two layers, the syncytiotrophoblast and the cytotrophoblast. The embryoblast differentiates into the primary germ layers of the embryo. The cytotrophoblasts are cells derived from cellular trophoblast and surround the entire structure. The syncytiotrophoblasts are finger-like projections from the trophoblasts that invade the endometrium. After implantation, the embryoblasts differentiate into the bilaminar embryonic disc. Around day 8, fluid collects between the epiblasts and overlying trophoblasts, forming the amniotic cavity surrounded by a thin membrane lining called the amnion. Also around day 8, cells migrate to form the primary yolk sac surrounded by the exoalomic membrane, or Heuser's membrane. Small spaces begin to form between the projections. These are called lacunae. As the syncytiotrophoblasts invades, it envelopes portions of the maternal endometrial blood vessels and release enzymes that erode the vessel walls. This causes maternal fluid to fill the lacunae spaces. The syncytiotrophoblast also begins to secrete human chorionic gonadotropin, which stimulates the corpus luteum to produce progesterone to maintain the pregnancy by providing blood flow to the endometrial lining. By days 10 to 11, the space between Heuser's membrane and the cytotrophoblast becomes filled with loosely associated extraembryonic mesodermal cells. By 10 to 12 days, it is completely embedded in the endometrium. By days 12 to 13, the extraembryonic mesoderm splits into two layers, one coating the Heuser's membrane and the other lining the cytotrophoblasts. The outside layer that adheres to the cytotrophoblasts is called the splanchnopleuric layer. The inner part that adheres to the exoalomic membrane, or Heuser's membrane, is called the somatopleuric layer. The space between the two layers is the chorionic cavity. By days 12 to 13, a second wave of migration of hypoblast cells produces a new membrane that migrates out over the inside of the extraembryonic mesoderm, pushing the primary yolk sac in front of it. This new layer becomes the endodermal lining of the secondary, or definitive, yolk sac. The pushing of the primary yolk sac also causes splitting of the extraembryonic mesoderm, forming the extraembryonic coelom, also called the chorionic cavity. As the definitive yolk sac develops on day 13, the primary yolk sac breaks up and is reduced to a collection of vesicles at the embryonic end of the chorionic cavity. By day 13, the embryonic disc with the dorsal amnion and ventral yolk sac are suspended in the chorionic cavity by extraembryonic mesoderm. Around the end of the second week, the definitive yolk sac loses contact with the remnants of the primary yolk sac, and the bilaminar embryonic disc with its dorsal amnion and ventral yolk sac is suspended in the chorionic cavity by a thick connecting stalk. Around days 11 to 13, the cytotrophoblasts proliferate into the syncytiotrophoblasts, forming primary stem villi. By day 16, the extraembryonic mesoderm begins to proliferate and invade the center of each primary stem villus, transforming each into a secondary stem villus. By day 21, the mesodermal cord differentiates into connective tissue and blood vessels, forming the tertiary stem villi. After implantation is complete, the original trophoblast surrounding the embryo has undergone differentiation into two layers, the inner cytotrophoblast and the outer syncytiotrophoblast. Lacunae in the rapidly expanding trophoblast have filled with maternal blood, and the connective tissue cells of the endometrium have undergone the decidual reaction in response to the trophoblastic invasion. Late in the second week, defined cytotrophoblastic projections called primary villi begin to take shape. Shortly thereafter, a mesenchymal core appears within an expanding villus, at which point it is properly called a secondary villus. Surrounding the mesenchymal core of the secondary villus is a complete layer of cytotrophoblastic cells, and outside of that is the syncytiotrophoblast. By definition, the secondary villus becomes a tertiary villus when blood vessels penetrate its mesenchymal core and newly formed branches. This occurs toward the end of the third week of pregnancy. The terminal portion of a villus remains trophoblastic, consisting of a solid mass of cytotrophoblast called a cytotrophoblastic cell column and a relatively thin covering of syncytiotrophoblast over that. The villus is bathed in maternal blood. A further development of the tip of the villus occurs when, under the influence of the local low-oxygen environment, the cytotrophoblastic cell column expands distally and penetrates the syncytiotrophoblastic layer. These cytotrophoblastic cells abut directly on maternal decidual cells and spread over them to form a complete cellular layer known as the cytotrophoblastic shell, which surrounds the embryo complex. The villi that give off the cytotrophoblastic extensions are known as anchoring villi because they represent the real attachment points between the embryo complex and the maternal tissues. It is important to understand the overall relationships of the various embryonic and maternal tissues at this stage of development. The embryo, attached by the body stalk or umbilical cord, is effectively suspended in the chorionic cavity. The chorionic cavity is bounded by the chorionic plate, which consists of extraembryonic mesoderm overlain with trophoblast. The chorionic villi extend outward from the chorionic plate, and their trophoblastic covering is continuous with that of the chorionic plate. The villi and the outer surface of the chorionic plate are bathed in a sea of continually exchanging maternal blood. One of the critical features of the developing embryonic-maternal interface is the establishment of a uteroplacental circulation that serves as the medium for bringing food and oxygen to and removing wastes from the embryo. This is accomplished by erosion of the walls of the spiral arteries of the uterus and their modification so that, as the embryo grows, these arteries can provide an increasing flow of blood at low pressure to bathe the syncytiotrophoblastic surface of the placenta. specialized invasive cytotrophoblastic cells, migrating out from the anchoring villi, invade the spiral arteries, but not the veins, and cause major modifications of their walls by secreting a specialized extracellular matrix and displacing many of the normal cellular elements, such as the smooth muscle cells, of the spiral arteries. As a result, the arteries become wider, but the blood escaping from their open ends leaves at a much lower pressure than normal arterial pressure. The placental circulation and the environment that it creates consist of two phases. For the first 2 months of pregnancy, the cytotrophoblastic cells that invade the spiral arteries essentially form plugs that restrict the flow of blood from the vessels. The first maternal fluid that bathes the embryonic trophoblast is not highly cellular, and the oxygen tension is low. As a result, during the first trimester the spiral arteries contribute relatively little blood to the maternal side of the placenta. This creates a low oxygen environment for the developing embryo and one in which much of the nutrition supporting embryonic growth comes from secretions of the uterine glands that bathe the outer surface of the placenta. In early gestation, low oxygen and glucose levels are important for the development of the embryo. During this period, the placenta grows more rapidly than the embryo. Within the low oxygen environment of the early placenta, vascular endothelial growth factor is a dominant growth factor, and it promotes angiogenesis within the placenta. Throughout this period, the fetal erythrocytes contain embryonic hemoglobin, which is adapted to bind oxygen under low tension. Starting at 9 to 10 weeks, maternal blood begins to flow through the intervillous spaces of the placenta, and the overall environment for the last 2 trimesters of pregnancy changes to one that is more highly oxygenated and richer in glucose. The increased oxygen levels downregulate VEGF, and through these and other influences, the fetus now grows faster than the placenta. During implantation of the embryo, the stromal cells of the endometrium undergo a striking transformation called the decidual reaction. After the stromal cells swell as the result of the accumulation of glycogen and lipid in their cytoplasm, they are known as decidual cells. The decidual reaction spreads throughout stromal cells in the superficial layers of the endometrium. One of the functions of decidual cells is to produce a layer of extracellular matrix proteins that facilitate the inward progression of trophoblastic cells from the embryo. The maternal decidua are given topographic names based on where they are located in relation to the embryo. The decidual tissue that overlies the embryo and its chorionic vesicle is the decidua capsularis, whereas the decidua that lies between the chorionic vesicle and the uterine wall is the decidua basalis. With continued growth of the embryo, the decidua basalis is incorporated into the maternal component of the definitive placenta. The remaining decidua, which consists of the decidualized endometrial tissue on the sides of the uterus not occupied by the embryo, is the decidua parietalis. The placenta consists of a fetal and a maternal component. The fetal component is the part of the chorionic vesicle represented by the chorion frondosum. It consists of the wall of the chorion, called the chorionic plate, and the chorionic villi that arise from that region. The maternal component is represented by the decidua basalis, but covering the decidua basalis is the fetally derived outer cytotrophoblastic shell. The intervillous space between the fetal and maternal components of the placenta is occupied by freely circulating maternal blood. In keeping with its principal function as an organ-mediating exchange between the fetal and maternal circulatory systems, the overall structure of the placenta is organized to provide a very large surface area for that exchange. The originally broad-based body stalk elongates and becomes narrower as pregnancy progresses. The umbilical cord becomes the conduit for the umbilical vessels, which traverse its length between the fetus and the placenta. The umbilical vessels are embedded in a mucoid connective tissue that is often called Wharton's jelly. Contained within the mesenchyme of the umbilical cord is a population of stem cells that, because of their accessibility, make them a popular source of stem cells for use in regenerative medicine. The umbilical cord, which commonly attains a length of 50 to 60 centimeters by the end of pregnancy, is typically twisted many times. The twisting can be seen by gross examination of the umbilical blood vessels. In approximately 1% of full-term pregnancies, true knots occur in the umbilical cord. If they tighten as the result of fetal movements, they can cause anoxia and even death of the fetus. Occasionally, an umbilical cord contains two umbilical veins if the right umbilical vein does not undergo its normal degeneration. Approximately 0.5% of mature umbilical cords contain only one umbilical artery. This condition is associated with a 15% to 20% incidence of associated cardiovascular defects in the fetus. Both the fetus and the mother contribute to the circulation of the mature placenta. Half of the volume of the human placenta is occupied by blood vessels. The fetal circulation is contained in the system of umbilical and placental vessels. Fetal blood reaches the placenta through the two umbilical arteries, which ramify throughout the chorionic plate. Smaller branches from these arteries enter the chorionic villi and break up into capillary networks in the terminal branches of the chorionic villi, where the exchange of materials with the maternal blood occurs. From the villus capillary beds, the blood vessels consolidate into successively larger venous branches. These retrace their way through the chorionic plate into the large single umbilical vein and to the fetus. In contrast to the fetal circulation, which is totally contained within blood vessels, the maternal blood supply to the placenta is a free-flowing lake that is not bounded by vessel walls. As a result of the trophoblast's invasive activities, roughly 80 to 100 spiral arteries of the endometrium open directly into the intervillous spaces and bathe the villi in approximately 150 milliliters of maternal blood, which is exchanged 3 to 4 times each minute. The maternal blood enters the intervillous space under reduced pressure because of the cytotrophoblastic plugs that partially occlude the lumens of the spiral arteries and because of the reduction of the functional smooth muscle layer in the walls of these arteries. Nevertheless, the maternal blood pressure is sufficient to force the oxygenated maternal arterial blood to the bases of the villus trees at the chorionic plate. The overall pressure of the maternal placental blood is approximately 10 millimeters of mercury in the relaxed uterus. From the chorionic plate, the blood percolates over the terminal villi while it returns to venous outflow pathways located in the decidual, or maternal, plate of the placenta. An adequate flow of maternal blood to the placenta is vital to the growth and development of the fetus, and a reduced maternal blood supply to the placenta can lead to poor fetal growth. In the terminal, or floating, villi, the fetal capillaries are located next to the trophoblastic surface to facilitate exchange between the fetal and maternal blood. The placental barrier of the mature placenta consists of the syncytial trophoblast, its basal lamina, the basal lamina of the fetal capillary, and the capillary endothelium. In younger embryos, a layer of cytotrophoblast is present in the placental barrier, but by 4 months the cytotrophoblastic layer begins to break up, and by 5 months, it is essentially gone. The transport of substances between the placenta and the maternal blood that bathes it is facilitated by the great surface area of the placenta, which expands from 5 square meters at 28 weeks to almost 11 square meters at term. Approximately 5 to 10% of the human placental surface consists of scattered areas where the barrier between fetal and maternal blood is extremely thin, measuring only a few micrometers. These areas, sometimes called epithelial plates, are apparently morphological adaptations designed to facilitate the diffusion of substances between the fetal and maternal circulations. The transfer of substances occurs both ways across the placenta. The bulk of the substances transferred from mother to fetus consists of oxygen and nutrients. The placenta represents the means for the final elimination of carbon dioxide and other fetal waste materials into the maternal circulation. Under some circumstances, other substances, some of them harmful, can be transferred across the placenta. Gases, principally oxygen from the mother and carbon dioxide from the fetus, readily cross the placental barrier by diffusion. The amount of exchange is limited more by blood flow than by the efficiency of diffusion. Similar to gases, water and electrolytes are readily transferred across the placenta. The rates of transfer are modified by colloid osmotic pressure in the case of water and the function of ion channels in the case of electrolytes. Fetal wastes are rapidly transferred across the placenta from the fetal circulation to the maternal blood bathing the villi. Amino acids are transported across the placenta through the action of specific receptors. Vitamins, especially water-soluble ones, are transferred from the maternal to the fetal circulation. Steroid hormones cross the placental barrier from the maternal blood. Protein hormones are, in general, poorly transported across the placenta. Some proteins are transferred very slowly through the placenta, mainly by means of pinocytosis, uptake by membrane-bound vesicles in the cells. Of considerable importance is the transfer of maternal antibodies, mainly of the immunoglobulin G, IgG, class. Because of its immature immune system, the fetus produces only small amounts of antibodies. The transplacental transfer of IgG antibodies begins at 12 weeks and increases progressively over time, with the greatest rate of antibody transfer occurring after 34 weeks. For this reason, prematurely born infants do not receive fully protective levels of maternal antibodies. The transfer of antibodies from the mother provides passive immunity of the newborn to certain common childhood diseases such as smallpox, diphtheria, and measles, until the infant's immune system begins to function more efficiently. Another maternal protein, transferrin, is important because, as its name implies, it carries iron to the fetus. The placental surface contains specific receptors for this protein. The iron apparently is dissociated from its transferrin carrier at the placental surface and then is actively transported into the fetal tissues. The placenta, specifically the syncytial trophoblast, is an important endocrine organ during much of pregnancy. It produces a wide variety of protein and steroid hormones. Many factors can contribute to embryonic death and can be divided into embryonic abnormalities, inadequacy of the maternal environment, and external factors. Complex biological processes do not work perfectly all of the time. More than 50% of fertilized eggs do not develop to maturity and are spontaneously aborted. The quality or health of an embryo plays a major role in successful development and implantation. For example, during in vitro fertilization, low-quality embryos were less likely to implant into the uterus. This may be the effects of natural selection acting to favor embryos that are more likely to develop, implant, and reach term successfully. Some evidence suggests that the blastocyst may not trigger the normal maternal response of implantation mediators and immunomodulators. The earliest stages of human development are highly prone to error because of chromosomal abnormalities which may occur during the critical steps of meiosis, fertilization, and the early cleavage stage. After fertilization, embryos undergo cleavage at a specific rate. If there are chromosomal abnormalities, the rate of cleavage may be too fast or too slow. Many embryonic deaths are due to the embryo having the wrong number of chromosomes. Chromosomal abnormalities represent the most common category of abnormality in abortuses, approximately 50% of the cases. Fertilized eggs should have 46 chromosomes, 23 from mom in the egg, 23 from dad in the sperm. Many embryos have the wrong number of chromosomes, often 45 or 47, and many of these die in the womb. However, embryos without the correct number of chromosomes may reach full term and delivery, such as embryos with trisomy 13, 18, 21. Further, if these embryos are implanted, it will not develop properly and many will not survive and either miscarried or a stillbirth. The maternal environment is an extremely broad category of potential sources of harm. This includes factors like insufficient maternal progesterone, inadequate provisions of nutrients from an aged, degenerated, or inadequately synchronized endometrium, and uterine infection or inflammation. Impaired endometrial receptivity is estimated to account for 2/3 of implantation failure in women with repeat implantation failure with in vitro fertilization. Failed implantation is found to be associated with a high body mass index, smoking, alcohol consumption, and stress. Pre-pregnancy obesity affects the female reproductive system through abnormal menstruation, anovulation, and pregnancy complications. Smoking has been associated with lower implantation rates in women undergoing in vitro fertilization. In women who smoked for over 5 years, there were fewer oocytes retrieved and lower rates of implantation compared to non-smokers. In male partners, smoking negatively affected sperm motility, count, and increased sperm DNA damage. Alcohol has a negative effect on pregnancy and the developing embryo. Stress increases cortisol levels and may be associated with an increased risk of miscarriage. The ideal implantation site of the blastocyst is the endometrium of the posterior wall in the uterine cavity when the uterus enters an implantation window during about days 20 to 24 of menstruation. The fertilized embryo attaches and implants about 6 to 7 days after implantation. Successful implantation requires a high degree of preparation and coordination by the embryo and the endometrium. Abnormal sites of implantation are rare but can cause serious problems. Implantation that occurs at an abnormal site is called an ectopic pregnancy. Tubal pregnancies are the most common type of ectopic pregnancy. Although most tubal pregnancies are found in the ampullary portion of the tube, they can be located anywhere from the fimbriated end to the uterotubal junction. Tubal pregnancies are most commonly seen in women who have had endometriosis, earlier surgery, or pelvic inflammatory disease. Scarring from inflammation or, sometimes, anatomical abnormalities result in blind pockets among the mucosal folds of the uterine tube. These can trap a blastocyst. Typically, the woman shows normal signs of early pregnancy, but at about 2 to 2.5 months, the implanted embryo and its associated trophoblastic derivatives have grown to the point where the stretching of the tube causes acute abdominal pain. If untreated, a tubal pregnancy typically ends with rupture of the tube and hemorrhage, often severe enough to be life-threatening to the mother. Very rarely, an embryo implants in the ovary, an ovarian pregnancy, or in the abdominal cavity, an abdominal pregnancy. Such instances can be the result of fertilization of an ovum before it enters the tube, the reflux of a fertilized egg from the tube, or, very rarely, the penetration of a tubal pregnancy through the wall of the tube. The most common implantation site for an abdominal pregnancy is in the rectouterine pouch, which is located behind the uterus. Implantation on the intestinal wall or mesentery is very dangerous because of the likelihood of severe hemorrhage while the embryo grows. In some instances, an embryo has developed to full term in an abdominal location. If not delivered, such an embryo can calcify, forming a lithopedion or stone child. The location of the placenta is crucial for pregnancy, and an abnormal placenta location can cause many complications for the pregnant women and fetus. Low-lying placenta and placenta previa are located in the lower uterine segment and are associated with increased obstetric risks due to excessive blood loss. A low-lying placenta is near, but not overlying, the internal os of the cervix. A placenta previa completely covers the os of the cervix. Because the lower uterine segment has a reduced blood flow, the placenta can detach more easily. This creates a cycle of placenta detachment, vaginal bleeding, cervical shorting, cervical dilation, and contractions. A pregnant woman with placenta previa will require a cesarean delivery due to the risk of excessive blood loss. There is also the risk for preterm birth, with rates of 26.9% with a low-lying placenta and 43.5% with placenta previa. Invasive placenta refers to a condition in which the placenta attaches too strongly or invades too deeply into the wall of the uterus. This may result in the placenta not separating and delivering as it usually does following delivery of the baby, and may cause extra bleeding if efforts to remove the placenta are made. Accreta occurs when the placenta attaches too deeply into the uterine wall. Accreta doesn't directly harm the fetus, however it can lead to preterm birth. This is the most common type of abnormal placenta attachment. Placenta increta occurs when the placenta attaches to the uterine muscle. Placenta percreta occurs when the invasive chorionic villi reach and penetrate through the uterine serosa. Once an abnormal invasion is diagnosed, there is no real treatment except close monitoring. When the time for delivery comes, the fetus should be delivered by C-section with surgical removal of the uterus to prevent uncontrolled hemorrhage. If a pregnant woman presents for delivery with an undiagnosed placenta invasion, the placenta may fail to detach after delivery, leading to postpartum hemorrhage. The placenta may develop with an abnormal form or shape. A bilobed placenta occurs when the placenta forms into two roughly equally sized lobes separated by a membrane. The umbilical cord may insert into either lobe, of filamentous manner, or between the lobes. It is not associated with fetal anomalies, but may lead to first trimester bleeding, polyhydramnios, abruption, and retained placenta. Rarely, it may form with more than two lobes, termed a multilobed placenta. A sexcenteriate placenta occurs when one or more accessory lobes develop in the membranes apart from the main placenta body, possibly with a fetal vessel connecting them. This is a variant of a bilobed placenta. The communicating membrane connecting the lobes may or may not have vessels. A circumvallate placenta is an extracorporeal, annularly shaped placenta with raised edges composed of a double fold of chorion, amnion, degenerated decidua, and fibrin deposits. It is associated with poor pregnancy outcomes due to increased risk of vaginal bleeding beginning in the first trimester, premature rupture of the membranes, preterm delivery, placental insufficiency, and placental abruption. A placenta membranacea is a rare placental abnormality where chorionic villi cover fetal membranes either completely or partially, and the placenta develops as a thin structure occupying the entire periphery of the chorion. This may be associated with placenta previa and placenta accreta. The ring-shaped placenta is an annularly shaped placenta that is a variant of placenta membranacea. It can sometimes be a complete ring of placental tissue, but more often, tissue atrophy in a portion of the ring results in a horseshoe shape. The ring-shaped placenta can cause antepartum and postpartum bleeding, as well as fetal growth restriction. Placenta fenestrata is a rare condition in which the central portion of the discoid placenta is missing. Rarely, there may be an actual hole in the placenta, but more frequently, the defect involves the villus tissue, and the chorionic plate remains intact. At delivery, this finding may cause concern for retained placenta. With vasa previa, membranes that contain blood vessels connecting the umbilical cord and placenta lie across or near the opening of the cervix. Some of these blood vessels are located in the membranes that surround the fetus, in the area between the fetus and the opening of the cervix. When the membranes rupture, these blood vessels can be torn. As a result, the fetus may experience hemorrhage. Placental insufficiency, also called placental dysfunction or uteroplacental vascular insufficiency, is a condition where there is a failure of placental vascular remodeling, leading to a failure of placentation resulting in acidosis and fetal hypoxemia. The most common downstream fetal consequences of this condition include intrauterine growth restriction, prematurity, or, unfortunately, fetal demise. Placenta insufficiency creates a process whereby there is a progressive deterioration in placental functioning such that oxygen and nutrient transfer to the fetus via the placenta is decreased, culminating in a decompensated hypoxia and acidosis. This process leads to fetal hypoxemia that then stimulates a downregulation of fetal metabolic demands to preserve what nutrients are already accessible, thus resulting in intrauterine fetal growth restriction. It may be due to a reduction in blood flow across the umbilicus to the fetus, which can be secondary to increased umbilical placental vascular resistance. This increased resistance can be visualized as abnormal umbilical artery Doppler flow velocity waveforms and can be secondary to an abnormality of villi insertion into the placental membrane, a perfusion abnormality between the umbilicus and placenta, or a reduction in uteroplacental blood flow. It is associated with various obstetric disorders such as preeclampsia and intrauterine growth restriction, both of which predispose to preterm labor, a leading cause of perinatal morbidity and mortality, but the major etiologies that may lead to placental insufficiency are poorly understood. There are known associated maternal risk factors, which include preeclampsia or other maternal hypertensive disorders, maternal cigarette use, maternal drug use including cocaine or heroin, maternal alcohol consumption, primiparity, advanced maternal age, and prior history of delivery of intrauterine growth restricted neonate. Any maternal condition that can lead to a compromise in the fetal circulation puts the fetus at risk for placental insufficiency. Additionally, certain medications such as antineoplastic, anticonvulsants, or anticoagulants can interfere with fetal growth. Extremes of maternal body mass index, including maternal malnutrition, have also been associated with the development of intrauterine growth-restricted neonates. There is no known treatment for placental insufficiency other than delivering the fetus if it is at a viable time point. The placenta is a vital fetal organ necessary for a healthy pregnancy for both mother and fetus. Incomplete or disordered development of the placenta may cause fetal abnormalities or adverse events in the mother as well.